which, as I mentioned, is so critical, and it keeps us honest, because we need to take we have to have a responsibility to pals to do things correctly, not to jeopardize them, but also not to jeopardize the clinical trial process, and but yet push for trials. So, but understanding a little bit of why you have a 20-page consent, I think this will help inform you a little bit. But I don't know, those of you who have been in trials, you know those consents. Every time I look at them, it's no wonder people don't want to read them, but you have to read them. Anyway, Hristelina, I'm looking forward to hearing about ethics and informed consent.
Okay, can you see my slides?
Yes.
Are they in? Oh, no. Okay. So I also want to thank Terry and Richard and Piera and everyone in terms of organizing this symposium, but also the patients. I mean, we're here because of you. We want this event to be as enriching as possible. So my slides were in a large amount done by Terry.
I should say that they were in large part done by Nazim Atazi initially, and it's been passed down through generations of those of us who have given this lecture. So I don't want to take credit for something that wasn't really totally mine. Nazim had done a great job putting these together. So
yeah, so Terry, Nazim, and then I added a few comments from the IRB manager with whom I worked. I have a unique perspective because for 2 years I also was chairing an IRB board for a hospital. So I came to see how IRB boards review informed consent. That's probably one of the most important documents they spend time on before they approve or don't give green light initially to a study. So that's why I told Terry, I think this is something that I can tackle. So this is going to be a lecture in a little bit of human history and how unfortunately humans learn or don't learn and need to repeat some mistakes before we get it right. So bear with me. We'll have two major points of discussion. One is what are the ethical principles that guide medical research now, and also what is the informed consent process? What are the components of those, of this process, and what forms are there, and what are the responsibilities? So as I mentioned to you, humanity, unfortunately, fairly often goes through painful experiences before it can synthesize some principles and try to avoid mistakes. One of the biggest mistakes of the 19th century was, the world wars that occurred, and in particular, World War II was very gruesome. Probably way better than I, that a lot of population was, tortured and prosecuted and killed, among them Jews, Poles, Roma, mentally and physically disabled people were subjected to a lot of cruelties, unnecessary, heinous acts in the camps. In order to get some, justice back, there was a trial that happened in German city of Nuremberg. And that's why the code is known with that name, the Nuremberg Code. And then it's also known as the doctor's trial. So 20 doctors were tried for the experimentation in the camps. One of those doctors is here. His name is Karl Brandt. He happened to be a neurosurgeon by training. It's fairly accepted by historians that without his tacit approval, none of those would have, heinous acts would have happened. He got sentenced by hanging, and that's kind of one of the outcomes of the trial. As I mentioned, the acts were unspeakable. They were, mutilation acts, they were sterilization, they were, purposeful infection with typhus, with malaria and other diseases, poisoning, giving people to drink salt water, et cetera, you name it. So because of the deliberations that, the judges had to do during that trial, they came up with 10 principles that so that hopefully humanity doesn't ever live through this nightmare. So what are the 10 principles in the Nuremberg Code? First of all, research has to be voluntary, so voluntarity of consent. Second of all, the experiments have to be faithful and good for the society. There should be some previous knowledge stemming from, animal data before experimentation is done on humans. Absolutely physical and mental suffering has to be avoided, and death and disability injuries cannot be induced outcomes. Risk should not exceed the humanitarian importance of research. There should be adequate preparation in terms of facilities, and the actual researchers who are doing this should be educated and scientifically qualified. Human subjects have the liberty to stop participation at any time point. And finally, investigators should be ready on their end to also terminate the study at any time point should results cause injury, disability, or death. So these are the 10 principles of the Nuremberg Code. They were then used in additional guidelines. For example, there's a Declaration of Helsinki, which is guiding also a lot of research with many reiterations of this declaration. And then, unfortunately, this sort of history happened across the ocean, but that is to say, we on this side of the ocean are not protected from making mistakes. And so that brings me to the next slide, where some of you also know about this chapter in American history, where a small city in Alabama, it's not a city, I think it's a town of Tuskegee, was chosen for a natural history study of syphilis in about 400 poor and untreated African American men. The participants were told that they are being treated for something called bad blood. That was a very non-specific term to mean fatigue, anemia, and included sexually transmitted disorder, syphilis. They were offered in exchange for their participation free meals and burial insurance. But the big controversy with this episode of American medical history is that the study continued despite approval of a drug curing syphilis in 1947. And if you see here, that experiment continued until 1972. So 25 years after the discovery of penicillin and approval of penicillin for treatment of syphilis, the study continued, and those men went being untreated, which in itself led to also unspeakable human suffering in terms of, infecting their wives, giving birth to children with congenital syphilis, things that could totally have been prevented. Not only that, the way this study ended is also notable. It wasn't ended by the researchers themselves. What needed to happen, of course, those participants were not told that they have syphilis. I told you that the word that was used was bad blood, and nor were they treated for it. But the way the study ended was actually by a whistleblower leaking information about this study to the New York Times, and then the New York Times published information, an article on that, and that led to a massive, outcry in the community. And of course, this was conducted by a government agency, and this government agency should have and could have protected those people between 47, after 47. So that's kind of the sad chapter on this end of the, of the ocean. Of course, we need to learn from mistakes and we need to kind of implement better safeguards. So the better safeguards came in three shapes. The Beaumont Report, we'll talk about it. The Office of Human Research Protection was established, and the institutional review boards were established, and we'll talk what exactly they are. So the Beaumont Report was done in 1978. It has three major principles: respect for persons, beneficence, and justice. Each one of them has, additional elements. The respect for persons is through autonomy of making your decision, informed consent, truthfulness, and no deception. Then beneficence, that's a principle in medicine from the time of Hippocrates, when you should do no harm in the first place, and the benefits should outweigh the risks. And justice, reasonable procedures distributed fairly among the society members. So the institutional review boards, this is an ethical board, which is an independent committee from any kind of hospital association, hospital affiliation. I mean, they should have enough autonomy to decide independent of the hospital. It's governed by the Code of Regulations 45, and also the OHRP, the Office of Human Research Protection, is governing institutional review boards across the country. Every institution who is doing human research has not only one, but big institutions like, big university have at least several institutional review boards in order to handle all the research requests. There should be representation of, the members at large, not only of medical personnel and not only of physicians, but many other, professions, pharmacists, nurses, speech pathologists, different kind of medical specialists participate in the boards. And in terms of members of the public, we've had clergymen, we've had, lawyers, we've had engineers, different kind of backgrounds. People from different background can be members of the IRB boards. So in particular, the safeguards, as we mentioned, include informed consent, where enough information is disclosed to the participant in order to make a decision. That patient or person needs to be having good decision-making capacity. And finally, this has to be done without coercion and voluntary. The institutional review boards themselves are another ethical safeguard, because they make sure that the appropriate research, appropriate meaning the one that's safe for the patients, that's good for the society in general, is done. At the same time, the privacy, confidentiality, dignity, and welfare of the patients are protected. The risks are outweighing the benefits, as we mentioned, and that an informed consent form documenting the informed consent process is filled. And then there are other agencies like the NIH, the FDA. So what exactly is the informed consent? We'll go through a definition, what are the components, the processes, and what is the form, and then what are the responsibilities of both the people doing the research and the people participating in the research. So you will hear occasionally the word subjects, and that's, to mean patients who participate in a research study. So the definition of informed consent is that it's a voluntary agreement based on the adequate knowledge and understanding of relevant information to participate in research or to undergo a diagnostic, therapeutic, or preventative procedure. That is definitely based on information, comprehension, and volunteering. So that's what the informed consent is. When we talk about respect for the autonomy, if you remember one of the principles of the Beaumont Report, what does that mean? That means that a person has the right to decide for him or herself, that a person has the right to information that will help them make that decision. The person has enough time to decide without being coerced, and the person can grasp the information because it's presented in a language and in a level and a word selection that is easy for them to understand. In practical terms, when we talk about information, what is information? Number one, people need to know that this is research. This is not standard of clinical care. They need to know what is the purpose of research, why is it being done. They need to know how long they will be expected to participate. They also need to know what are the plans and what procedures are there that the researchers plan to do. And also they need to know if this is greater than minimal risk. What is there any compensation? When we say something is greater than minimal risk, any kind of new medication that's being studied is considered greater than minimal risk because, of course, it's a new medication. We don't know all the possible, side effects of that medication. So then, is there some sort of compensation available? And then, of course, they need to know that their participation is voluntary and they can withdraw it at any time point. And also, who do they ask questions from? All of these elements are actively being looked for by IRBs when researchers submit the consent form. Is that information part of the IRB consent form? Some additional elements that are also being carefully looked at is the mentioning that at this point in time, researchers, clinicians may not know of some risks. So there may be risks that we are unaware of that should be stated. Also, it should be stated under what circumstances it's possible to ask a participant not to continue in research. And those circumstances can be discussed. Also, in some cases, there may be additional costs associated with the participation. That should also be mentioned. How many people are expected to be enrolled in this project, in the study? And also, if there are new findings, now that will ring quite familiar with the Tuskegee experiment. If there are new findings that become available during the research period, how would that impact the participants and their willingness to continue and to share that information, the research information with them? So informed consent is not a document. That's what some, people, on first pass think. It's a process. It's a continuing dialogue, which begins at the time when the researcher or the clinician mentions to you there is a study. So that's the beginning of that consent process. There should be, as we mentioned, sufficient time to review the consent and to answer the questions. Exactly what Terry mentioned before. Some people, they've made up their mind. They already know they want to do the research, but nevertheless, we really want the participants in the study to read what is there in the informed consent. And we oftentimes have the practice of sending you the informed consent ahead of your, time when we'll be actually meeting you in the research setting, so that you had ample time. You could consult, a dictionary if there is a word that bothers you, et cetera, et cetera. And it's dynamic. It's not like a one-time situation that is written in stone and never changes. No, it's, a process. It can go through ups and downs. People can, refuse or stop their participation, and that's fair. Finally, can you hear me? Okay. Finally, when does it end? It ends with the post-study communication when people share results with you. So what about comprehension? Subjects must be able to understand the information that's presented to them. And usually most IRB levels have a set of words or, that are accepted as easy to understand, common language. That's usually 8th grade level. Information must be adapted if, there's some word, as we said, or expressions that are difficult to understand. And then there must be enough sufficient time, not only to read the actual document, but also to have this dialogue, to have the back and forth answers and questions. Also, the other important part is that there should be no form of coercion. There should be no physical, no psychological or monetary coercion. Sometimes IRBs spend a lot of time discussing if there will be a compensation for a study. Is that reaching the level of coercion? If that compensation is significant, it may be viewed as a way or form of monetary coercion. So that's another aspect. Another, how do we actually show our respect to people when we go through informed consent? Well, first of all, the person understands what's being asked of him or her. Also, the respect is if the person has the opportunity to ask questions. And finally, the person truly wants to continue with the study. It's not something that he or she does because they want to please their doctor. No, it shouldn't be that way. So what are the basic components? We a little bit talked about those: the purpose, the visits, how many visits, what are the procedures, what are the potential risks, what is the animal and the human data, what are the common side effects, what are the rare serious side effects, the potential benefits. There may be no benefit to a participant since the treatments are experimental by design, and then we should be honest with people and say that. And we always learn something new. And if that's the case, then it may not benefit the current participant in the research, but it will benefit people, patients, families, communities in the future. And then, for example, are available current approved treatments approved under, to be taken under the particular disease group, under the particular disease we're studying? For example, is riluzole, radicava, BiPAP, etc. allowed? Most studies do that. They do. It's unethical not to allow people to use FDA-approved studies, medications, and procedures. Then, if there's new information, if something new comes, then we need to inform participants of newly approved treatments. The confidentiality is very important. People get, specific code of letters and numbers such that their first name and last name is not used anymore. Then how is that data stored is important as well, and who has access to this data. We mentioned a little bit about compensation for time. There's also specific language that lawyers are very careful about in terms of medical injury. And then we also mentioned that compensation has to be very reasonable, not to present, a form of monetary coercion. And finally, what is the contact information? What are the questions about, the study? If you have research questions, etc., who do you contact? How do you contact? So all of these sets of data are included. And finally, assuring voluntariness, that you will not lose any benefits. You will not lose your autonomy if you decide not to participate in a study. So the actual form is that legal document that meets all of these elements that we just talked about it. It's kind of like a reminder for you when you go home, you can reread them, you can discuss with family, and it serves you as kind of a basic guide. It provides the documentation and proves that, that sort of conversation happened. It's signed always. Not only that, it's dated, and a lot of times we also put the actual time of the day when it was signed and dated, that it was sometimes witnessed as well, and then copies were given to you as soon as this was done. What about the responsibilities? Safety is the biggest responsibility when we participate in clinical trials. Our biggest concern is, are we doing enough to keep you safe? Ensuring that the patients their rights are protected. We talked a little bit about the safeguards there. And also we have to be compliant with all the laws and regulations that exist, federal, state, and locally mandated laws. So in terms of your responsibilities, for you, it will be important to take as much time as you need to understand all the implications of the research. What exactly is the treatment? What is the benefit risk ratio? Is this concurrent with current medical thinking, with current guidelines? And is the investigator and their staff trained to perform this sort of investigation? And are your questions handled in a way that's satisfactory to you? Are things explained in a way that you can understand? So there are ethical safeguards. We mentioned the major ones, the Nuremberg Code, the Beaumont Report, and the current, safeguards of any research done in this country are the Office of Human Research Protection, the Institutional Review Boards, the Food and Drug Administration. The informed consent is a process in which information, comprehension, and volunteering are the basis. It's a dynamic and ongoing process, and there are shared responsibilities between the patients and the investigators. So thank you very much.
Thank you, Doctor. That was an awesome presentation, and we really appreciate you putting that together. It's really informative, and really, it really helps the patients know what to expect. So thank you.
Thank you.
We do have a couple questions for you. First question is, can patients and family members be on an IRB, and how, if so, how can they apply?
I think that patients and family members can potentially express their interest. Usually IRB members rotate on a certain time principle. Usually it's either 1 year or 2 years. And if there's an opening, they can certainly voice their desire. That will be very helpful for the IRB group because, I'll be honest with you, these are non-compensated, jobs usually. So you're given homework to do to prepare for an IRB committee meeting, which, depending on the frequency, happens anything between, once a week to once every 2 weeks, depending on, the frequency. But you will have to then read the informed consent of a certain study, try to understand it, try to ask the questions that you will ask as, a person that, is being asked, for example, to do this research and help the institutional review board in that way to, flush the uncertainties or the questions that arise. And then you'll do the homework and then you'll go and have the actual meeting, which usually takes 2 or 3 hours. So it's not something that you can just participate once. If you commit, you should take it responsibly and do it for about a year, because it's a learning curve. You don't know initially enough. You will then learn enough to feel comfortable, and then, it becomes a lot easier. But it is a involved process.
I believe it or not, when I work for Ascension Hospital and I volunteer on the IRB. So I am an active IRB person.
I'm very glad. Thank you.
Yeah.
Hristelina, I would think most of the IRBs, have a website and a phone number.
Mm-hmm.
And I would think that interested patients and family members, could reach out to the IRB through their website or their phone number and just say, if you're looking for a new community member, I'm interested in volunteering. I just think, like I mentioned before in my talk, it's so important that these regulatory groups understand this disease, and there's no better way they can understand it than by interacting with people and families affected by the disease.
100% agree. So if there is anyone interested, in volunteering, in particular IRB close to your home, close to where you go and are evaluated, please reach out to them and ask that question. Yeah, I think it's a very good point. Thank you, Dr. Bedlock.
Great.
Remember
Uh,
that, I was just going to say that, remember that you will be reviewing not only ALS protocols, but all
Mhm.
kinds of research in that
Mhm.
context.
Doctor, if a DSMB has an adverse event, either for a serious or lethal, how do informed consents deal with that, and how much info are participants given about the adverse event?
So if I understand the question correctly, it's asking if an adverse event happens during a trial, will that information be part of the informed consent? Is that sort of the question?
Yes.
I mean, I think.
I'm sorry, it was my question. I just didn't know if I'd be able to unmute. I just wanted to know how the informed consent addresses that ahead of time.
Right.
And then how much info is given back to a patient if there is an adverse event when a trial is put on hold, or does it vary with the consents, with the informed consents?
I mean, as we mentioned at the very beginning, if this is new information, let's kind of play it in two ways. If it's known information, it will be included in the possible adverse events. And usually the IRB boards want to see, frequent adverse events versus infrequent adverse events. So if this is something known, this particular, adverse event will be included as a possibility so that whoever reads the consent can, definitely be informed about this possible adverse event. Now, if we are talking about a new type of adverse event, it will not be included in the informed consent. So then.
But I'm sorry, would the parameters under which patients get advised be included in the informed consent? That's what I was trying to figure out.
I am not following the question. When you say the parameters, what do you mean?
If the informed consent has language in it that says if there's a new adverse event that comes up that we didn't anticipate, what does the patient get notified of, and how does the informed consent anticipate future adverse events that may or may not happen from a DSMB review?
Yeah, so if you remember, I had a point in information where I mentioned that we have to tell people that there may be risks that we are currently unaware of. So this is the kind of blanket statement that a lot of the informed consents will contain, where they, kind of say, it's possible we don't know all the adverse events or all the things that can happen. So, be prepared for something like this. But that informed consent can go, through revisions. And so if something is observed, then, and this is, something that needs to be included, then once this informed consent is revised, what I'm trying to tell you is that the initial information may not be there, but as revisions of the informed consent happen, and they happen periodically, we need, new informed consents because, there's only a certain period of time under which the informed consent is approved. If this new event happens and is included in the next, it will be supposedly included in the next revision of the informed consent form. But that doesn't mean that we're talking about the form, but as we spoke, informed consent is a process. Even if something is not included verbatim in the form, still the investigator may, communicate that sort of event in their conversation with patients. Is that answering your question?
Yeah, I just a little bit. I'm trying to understand what a patient gets to know if there's a hold as to the why. Like,
Yeah, but
you know,
have you
what are they
seen,
told before they,
have you,
before
yeah,
they go on?
that brings me to my question to you. Have you seen an informed consent?
Uh-huh.
Okay. And then have you seen it for a research, for a, you know, a drug, for a therapeutic drug?
No, because I've not ever participated in a trial, but
Yeah.
I'm on the other side with the patients that when Dr. Bedlack, I know you were on the DSMB for a lot of boards, so that's kind of what I was trying to gather. When an adverse event happens, what does the patient get told about the adverse event in the informed consent? How much do they know to make the decision about whether they should continue in a trial if there's an adverse event? That's kind of where I'm trying to get to.
Dr. Bedlack, do you want to take that one? Because it was addressed to you.
Yeah. So, I mean, what we start off with is all the bad things that we know that can happen with the drug. And the problem is when it's real early in development, we, as Hristelina said, we might not know everything bad that can happen. You know, like if it's a phase 3 study, well, it's already been around. We have a pretty good idea of what bad things might happen. But if it's phase 1 or phase 2, we may not know. And so we put down what we know, and then, you know, as new adverse events and serious adverse events come in, DSMBs review those. You know, the first thing that happens is the investigator of the site gets the adverse event or serious adverse event about their patient, and they have to look at it and say, do I think there's any chance this could have been related to the drug or to the protocol? And, you know, then the DSMB overreads all that. We look at everything that comes in and we say, do we agree with the investigator's assessment? Because we've got sort of the, you know, the view from above. We see all the sites, not just what's happening at the one site, which is what the investigator says. And there have been times when a DSMB says, hey, there's enough of these things coming in that maybe this is related, and we probably do need to change the consent form. So if it's just a random thing that happens here and again, and it seems like it's unrelated to the drug, no, that doesn't get in. But if there start to be a few of them, and the DSMB is concerned that maybe this is related, then yes, there's a recommendation made that the consent form be changed. It goes back to the sponsor. Sponsor typically agrees and sends out, you know, the next version of what they think the informed consent should read. So we try our best to keep up with things that are coming in.
Is it fair to say that as a general term in the informed consent is written there is written that in case of severe adverse effects, there will be a review and eventually the trial will be stopped? Or the is that what you were looking for, Michelle? Just the general term in indicating that, again, because as it was said, there is no way to know in an investigation drug, right?
Right.
What the adverse effect will be, but as a general thing, saying that those effects will be monitored throughout the trial, and therefore, should they arise, there will be an investigation, and as Dr. Bedlack said, and eventually that will be addressed.
The thing that made me ask the question was in the Mayo mesenchymal stem cell trial in the phase 1, somebody fell and they put a hold on the trial. And, you know, if you're a patient and you don't know why there's a hold on the trial, that obviously causes a lot of concern for you as a patient. And so what does the patient get informed about that hold, and when it's, you know, it's just like, how much information do we get compared to what's unblinded to somebody that's on the DSMB, and when? That's kind of the context of the question.
Yeah.
Because it ended up being nothing, right, in the Mayo trial. Somebody tripped and fell and hurt themselves, and it wasn't related to the treatment, obviously.
Yeah, I mean, I don't know the details of that particular trial, wasn't part of that one, but there have been times when we have, as a DSMB, raised concerns about something that we saw once or twice, and, you know, we weren't sure. And what we typically do then is we typically suggest a letter to the investigators at the sites. And then the investigators are, you know, expected to share that information with patients. But when we see it enough, we request that the consent form be changed so that we know that people are getting a certain verbiage when they sign the consent. But it's a whole process. I mean, it is, as Hristelina pointed out, we do try to put the words in there that we may not know of all the bad things that can happen. And this is exactly why. But I think in
And
general, DSMBs err on the side of stopping or err on the side of warning people, maybe earlier than they need to, because we are most concerned about safety. That's what the S stands for. Safety is our primary concern when we're on those committees.
Can I just add there's one other layer there. So any investigational product has an application to the FDA. So in addition to the independent data safety monitoring board, any serious adverse event gets reported in real time to the FDA if it's considered to be study drug related or study related procedure. And in the mesenchymal stem cell study, the procedure is an injection into the spinal, into the intrathecal space, into the spinal fluid. And so until there's a determination that it's not related to the study procedure or the actual investigational product, even the hold may come from the FDA until all the data has been reviewed, especially in early phase clinical trials. This happens less so in later phase clinical trials because you have some more understanding. But there is, and usually in sponsored clinical trials, so where there's a pharmaceutical company, there's not only an independent data safety monitoring board, which includes specialists that understand the disease and can put context to any kind of adverse event. So that's the independent. There's also an unblinded safety or pharmacovigilance team that's not only surveilling the actual clinical trial in ALS, but that product and any other indication across its lifespan. So that's happening in the background.
The last thing I will add is, Michelle, any of those documents are very carefully, extremely carefully looked at by lawyers. So the language, that you will see has been, massaged and very carefully selected so that, they serve the purpose, what, they need to serve. So I don't think that it's very easy, for one event to automatically be reflected into the informed consent. So as Dr. Bedlak said, it takes a little bit of time. But if there is something, then they will act. And then, as we spoke, it doesn't have to appear in the written form. If a researcher receives that letter, then I think he has the freedom to share that information with their patients. Yeah.
So Michelle, were you also asking what, for just as an example, if there's a 12-month study and in month 10, there are some adverse reactions, and is there a timeline in which patients who are in the study, should be told, or is that process like months, or, how much information and in a timely fashion are they given? Is that kind of what I'm understanding correctly?
I can answer that. That was my job actually in industry. And so what I can tell you is that as each especially serious adverse events, this means that it either had a major medical impact, it resulted in a hospitalization, or it resulted in death, it was life-threatening, or it caused fetotoxicity. So that classifies any kind of event, a serious adverse event by regulations. If something occurs, usually it's reported within 24 hours. The job of the investigator is to report it within 24 hours to the sponsor. Sponsor has to determine causality. And so an unblinded team who knows the product will look and review based on the PI's input and their own knowledge of the drug, determine whether it's related or unrelated. They have to make that determination within 7 days, or 7 to 15 days, 7 days if it resulted in death. Now, and this is true in Europe and in the US, if it is related, it's considered a serious unexpected serious adverse reaction, SUSAR. And so this requires reporting to the FDA and any other regulatory authority in real time, and a report is usually generated to the site. So as these serious adverse events occur, a report is generated to all the participating investigators to review in real time. And these have to be submitted to their ethics committees or institutional review boards as the study is ongoing. So if there's something that looks really significant, like sudden unexpected death, that's something that needs to be discussed. And if it is something significant and related, it does alter the ICF at that time, and an amendment has to occur. And for anyone who participates in clinical trials, you sometimes have to reconsent. So sometimes if the study procedures change, the safety risk change, that would require someone to sign a new consent form.
Oh, we can't hear you.
Michelle, you're muted.
I asked is as people come up with delay in diagnosis and they come up to their 2-year time frame or their 3-year time frame, if there's an adverse event in a trial, they may decide, I don't want to stay in the trial. I may want to try to go find a different trial while I still qualify. And so that's why I was trying to get a sense of how much are they told about the reason for the adverse event, so they can make an informed decision about whether they want to stick with this trial or maybe move to another one while they still can.
That's a common occurrence. I've had that happen where people have a serious adverse event, and it could be due to an array of things, whether it was, and at the time, I'm not entirely sure whether it's related or unrelated, but it was serious and it caused a lot of burden for the person participating in the clinical trial. At that time, some people will stick with it based on, whether they really feel that that it wasn't really related, or they really have a lot of hope in the mechanism, or they're maybe in a later phase clinical trial with some established efficacy, or they might choose at that time to discontinue. And yeah, the decision is hard when you're not entirely sure whether it's drug related or not, but it's a personal decision based on what actually happened, their medical history, and what it entails continuing in the study, because sometimes there's windows. If you miss the next dose window, for example, it might not be possible if you're hospitalized for a prolonged period of time to continue in a study. And then usually, depending, most investigational products, you're able to discontinue, wash out, and then try in another different clinical trial.
Are there any other comments or questions? Thank you, Jinsey. It's really helpful to have you here. Thank you.
Terry, just one comment. Hristelina did a great job with that. The reason I wanted this lecture to be in here, and the reason it's been in every CRLI, is because one of the main reasons that I hear patients say no to a clinical trial is they say, I don't want to be a guinea pig. And when I ask them, can you explain what that means? They say, I feel like if I sign that piece of paper, you're going to be able to do whatever you want to me. And I might not always know. And so now when you go around to speak to people at support groups and clinics, and they're not participating, if they give you that answer, you can see why they feel that way, because that actually happened in the past, including right here in the United States in Tuskegee. But you can also see why it can never happen again.
Yeah, one comment I was going to make is, if you think carefully about what the Nuremberg Code said, number 10 was about, the need of the, new information to be shared, right? Investigators should be ready to terminate the study at any time if continuing will result in injury, disability, or death. That's exactly what, happened in Tuskegee. So the point I want to make is that we can put all the words that we want in law in books, but it's the responsibility of us as members of society, as responsible, investigators, you as patients, to safeguard each other. Because the sad thing is, if it happened once, it can happen again. And even though you can write all the laws and all the books and have it written down, it just comes down to practice, good people helping each other and making sure, as Dr. Bedlack said, that there's education, that there's, information that is, shared with people. That's the only comment I will make. I think there was something in the chat. Okay. I thought there was another question there, but yeah, that's my only comment. And
Any other comments or questions?
I just want to say real quickly, this is Brian, and Hristelina is the one who's running my care down Jefferson. And one of the nice things about this whole thing, I'm going to be honest, as a patient, your head is spinning, and you've got 75,000 million things on your mind. So to have somebody like Hristelina and her team doing that all this work behind the scenes so that you don't, I mean, I don't mean you don't have to as a patient. You need to be your best, your own advocate, but it's really comforting to know how serious you all, Terry, all you guys take this, because again, you're the one doing all that work. We don't have time. Not that I don't have time for it, but you don't, and so when you have a doctor who is being that meticulous about it, it is extremely comforting as a patient. You can enter that trial knowing you are in the great hands of somebody who is, again, meticulously reading all those things. So I just want to say it's really important. It puts your mind at ease, really does. That's it. I just want to say that.
I
I
cannot,
couldn't agree more.
thank you.
Thanks for those comments, Brian. I think all of us who do clinical trials feel that way. We may joke a little bit about the size of our consents, but we all feel a huge responsibility to keep you guys safe.
Yeah, no, agreed. It's
Do no
very,
harm,
very
first
comforting.
tenet. Do no harms, first tenet of medicine, no matter what you do. So, well, very good. So we, I think, are going to have a little break, another bathroom break. Lyle is probably going to join us right at 4:30 after his other talk is over.
I'm here.
Oh, are you here, Lyle?
Yeah,
I
he's
showed
here.
up just in time to hear wonderful things about Hristelina, which I fully endorse and agree with. And Hristelina, I haven't seen you in a while, but I give you a hug, and she
Hugging
was
you
wonderful.
all.
Yeah, we miss you at Hopkins too. So.
Well, very good. So Lyle, did you want to go right into your talk then?
We can take a break. It's fine. Whatever.
What would people like to do? Do people want to take a little stretch and come back at 4:30? What? Yes, I see some yeses. Okay, so why don't we come back? Everybody sort of come back at 4:27 so we can be ready to go at 4:30. I could use a break myself.
It's 4:24 now, so why don't we say a 10-minute break?
Oh, is it?
That's
I
fine.
thought it was already, it
Yeah.
was only 4:15. Okay, good. So
Yeah.
4:30. What were you saying, Lyle?
I was saying maybe at least a 10-minute break for people
Yeah,
to stretch.
10 minutes,
and get-
4:30, 4:35.
Because I
That's
need you
okay.
awake for the next talk. I need everybody ready to go. So go, get psyched.
Todd, where are you from? Even though I just put you on the spot. There you go.
Northeastern Pennsylvania, a little town called Brackney.
Brackney, okay.
It is probably 60 miles north of Scranton
Oh, wow.
and about 7 miles south of Binghamton, New York.
Okay, so you're way up there.
I'm way up there.
All right.
I'm not close to anything.
No, I would, yeah, I hear you.
So when I come down to Philly, it's a trip. We come down, it's a 3.5 hour drive, maybe.
Okay,
Yeah.
it's a long one.
Yeah.
Is that close to Sayre at all?
Sayre is about 30 to 40 minutes west of us.
Okay, that was my birthplace.
Oh, yeah.
Yeah.
Yeah, I mean, it's a nice little town, but we don't get over there too often. There's not a whole lot there, really.
No, not much to do.
No.
Well, we're glad you can join us. I think you're our furthest north person. I thought Sarah Brendel might be north, the furthest north, but I think you got her beat.
Definitely. Are you on my way to Syracuse though?
Yeah, I'm maybe, like I said, about 7 or 8 miles off of 81. So if you're going to Syracuse, you go up the turnpike and you'll hop on 81 and follow that right into Syracuse. You'll zip right by us.
I've only been there once so far, but my son just committed to Syracuse, so
Oh,
he'll be
boy.
starting there in the fall. So we'll be driving right past you quite often.
There you go. If you're ever in a jam on the road, that's where you're going to be.
All right, we'll have to connect.
You get up there close to Syracuse, the weather can change in a heartbeat.
Yes, yeah. He's tired of everybody telling him he's going to be cold.
I actually have a buddy whose stepson just committed to Syracuse too, but
Cool.
yeah, they get in. That's just a snow belt right there. They get, sometimes the wind blows the wrong way off of Lake Ontario and it just buries them.
Yeah, I'm excited for him.
Yeah, I mean, everything's got its pros and its cons, you know.
Right.
Sarah, what's he going to study? Does he know yet?
Yes. Wait, I have to make sure I get it right. It's broadcast and digital journalism.
That's it. That's the place to go. They're the #1 school for that, isn't it?
Yeah, and he got really lucky. I mean, he worked his butt off. Don't get me wrong, he earned it, but they're giving him a very nice deal. So
That's great. Congratulations. You must be
thank
proud.
you. We are.
Yeah. All right, I think we're going to start in 1 more minute.
Yeah, we'll give people a little bit longer. Lyle's all queued up.
You guys can see my screen, right? I hope.
Yeah.
In presentation mode, not in some goofy thing, because when I just went to go give that other lecture, Zoom would not give me my microphone back. It was a real hassle. They use different software and I had to like reboot.
I think they actually paid someone to run it.
Dr. Terry, are we during the social hour? Are we allowed to have a alcoholic beverage as well?
Absolutely.
Yeah, I just want to make sure.
We got to check your ID.
Yeah.
Woohoo!
Hell yeah.
The question is, can you find Grubhub to deliver you an alcoholic beverage, right?
We'll try to find a way.
Yeah.
I laughed when I saw that Grubhub thing. I said, well, we'll have to hold on to that till we go back down to Philly because there isn't anything up here.
Is that right? Is that right, Todd?
Oh, no, I mean, there were some people when everything was shut down, they were branching out a little bit, but I don't think they'd come to my house.
Where do you live, Todd?
Brackney, PA. It's a no-name little hole in the wall, but we're just south of Binghamton, New York, and about, I don't know, it's maybe about 50 miles north of Scranton.
Oh, wow.
We're
I didn't
way
know
up.
there was anything north of Scranton.
Oh, there's lots of the beautiful stuff's north of Scranton.
That's right.
Actually, whoever Sarah was saying, when she goes up to Syracuse, she needs to branch a little bit west and check out the Finger Lakes. It's gorgeous up there.
Ooh, that sounds good.
Wine country.
Oh, Grubhub? Grubhub wine?
There's Grubhub in Binghamton, but they won't come to us.
Oh, gosh. Well, I'm sorry about that. We were just trying to, normally if we were in person, put you up in a nice place, and we have some really great meals and a lot of time after the coursework to go to the bar and hang out and and
yeah, well,
know each other.
We're in Philly at least once a month for treatment, so we'll be as long as the open label keeps going, we'll be there once a month.
There you go.
There you go.
Great. So, Lyle, do you want to go ahead and get started?
Yep. You guys ready? Okay. So we're going to talk a little bit about biology and how do we decide what it is that we're going to study, and how do we decide what might be involved in this disease. Usually when I give this talk, it's a little more interactive, and I get to get you guys on your toes and pick you out of the the audience and quiz you a bit, but it doesn't really work so well with the Zoom, so you're kind of off the hook, but maybe play along anyway. And so these are the things we're going to talk about. And when today started, I was sort of struck. I mean, Rick, when he gave us the introductory talk, he talked about when he was a resident, right? And the first patient that he met, and his attending, I think he said something like, told him that, we know what this is called, but we don't know why it happens, right? And, Piera then talked about all the different kinds of ALS and the need for personalized medicine, because sporadic ALS is so complicated and there's so many different flavors and different things that seem to go wrong, and the science is so complex. And when you guys were introducing yourselves, I think, was it Sarah, you said that, they think you have PMA, right? And I think Beth has PLS, right? Did I get that right? And then, Chuck mentioned that originally somebody said MMN, and then that changed to ALS. And so, what does that all mean? Like, what actually is ALS? What do we mean when we say ALS? So we're going to talk about that, and actually how we diagnose it, and maybe what causes sporadic ALS, and then again, how does that inform what we look for? What sorts of biology are we interested in? And I think Jinsey's giving a talk later, right, about clinical trial, clinical trial design and challenges. I don't have to tell you guys, like, we have a bad record in this disease, right? Like, we have all this amazing basic science, and it sounds so cool, and all this stuff that we figured out in the lab, and we've not done a very good job of what Piera was talking about, which is bridging that valley of death. That's the translational piece, right? And so why? What is it that, why are we having such a hard time translating things? And I think most importantly, when we think about these questions, how can this help us think about how we can do better? And why are we optimistic for the future? So question one is, what is ALS? And in the simplest sense, so ALS, ALS is a clinical diagnosis. It's a disease of progressive weakness, and the weakness can start anywhere, right? It can start in your hand, it can start in your breathing muscles, it can start in different places, and then it spreads from there. And in general, there's certain muscles that are spared. We don't quite understand why, like eye movements, bowel and bladder control. There's ALS is a fatal disease, as you guys well know. And the reason it's a fatal disease is not because your arms become weak, it's because eventually it involves the muscles of breathing, right? And the muscles of swallowing. And some patients also get this kind of dementia, this frontotemporal dementia, which is more common with some of the genetic variants. But really, that's the definition of ALS. And so aside from the cases that are due to some of the genetic mutations, I think we have some people on the call who are C9 and SOD1. We don't have a blood test to confirm a diagnosis of ALS, right? We don't have a way to confirm it. It's a clinical diagnosis. This is a book I have from, it's 100-year-old. I like medical history, but it's got a really, I think, a very good picture drawing of, this is the system that we're talking about. So what's called the cortical spinal system. And it's called that. So this is a slice through your brain, like this way, right? And so this is your brain, and this is your spinal cord, and the brainstem that connects them. Can you guys see my arrow or not? Do you see my arrow? Okay. And so, the outer part of your brain is called the cortex, and there's a part of the cortex that controls motor function. And it's got this, you guys have probably seen it, something called a homunculus, where the face is controlled by one place and the arm and the leg by another. And then all those fibers, they come together and they go into this little pathway and they go down the brainstem and into the spinal cord. And then once they're in the spinal cord, they touch other neurons. So this is the upper motor neuron, and they touch the lower motor neuron, and that forms nerves that then go out to your muscles and make your muscles move, right? And so, nowadays you can just Google, like, and so this, by the way, that's why this is the cortical spinal tract, right? That's the motor tract there. You can just Google motor pathways and you get these fantastic images from anywhere. They don't actually show much more than that one I just showed from 100 years ago, right? It's the same thing. It's showing that these are different. This is the strip that's called the primary motor cortex. This is outside of a brain. This is the front of the brain, the back of the brain. And so this person will be looking over this way. And the tissue, the nerves from up in there, they come down and they come together and then they cross over, they go down the spinal cord. Spinal cord's got a lot of other stuff in it, right? I mean, things for sensation and all the different kinds of stuff that your body does and your brainstem and everything else that our brain does. And yet somehow the only thing that really is affected, significantly in this disease are these motor tracts. So how does a neurologist diagnose ALS? Well, when a neurologist sees a patient for the first time, and actually whether it's ALS or something else, the first thing we're doing is we're not trying to say, what disease do you have? We're not trying to say, do you have ALS or multiple sclerosis or Parkinson's disease or a tumor or a stroke? What we're trying to do is figure out where can we point our finger in your nervous system that might be having the problem, right? If somebody has weakness or sensory loss or they can't see or whatever it might be, we're trying to figure out where in the nervous system might the problem be. And then once we figure out where the problem is, we think of the kinds of diseases that can affect that area of the nervous system in the sort of tempo and pattern that we're seeing. So this is my scribbly picture of those really beautiful pictures that we just saw. Again, brain, spinal cord, you got nerves up in the brain from the motor cortex, they come down, they touch other nerves, they go out to the muscles. You can have weakness because of a problem anywhere along here. So if you've ever known somebody that had a stroke, maybe they had a stroke up in their brain and maybe they couldn't move some part of their body, right? Or someone that has an injury to their spinal cord, or somebody that has a neuropathy, like somebody with diabetes and maybe can't move their toes. There's diseases of the connection between the muscle and the nerve. This is something called myasthenia gravis, where this signaling is screwed up and those patients have weakness. Or muscle diseases, right? So, people who take cholesterol medicine, some of them get something called myositis, or children with muscular dystrophies. And so a problem anywhere along this path can make you weak. Now, if we draw a line through this pathway right here, and so we separate out the brain and the spinal cord from the part that's this lower motor neuron and the connection in the muscle, we see different things on exam if the problem is on this side of the picture or this side of the picture. And that's, you've probably heard your doctors refer to, upper motor neuron signs, which would be problems on this side, or lower motor neuron signs, problems on this side. And so the upper motor neuron side, spasticity, increased reflexes, emotional lability, these are things that would suggest to us that there's a problem on this side of the picture. Lower motor neuron sides are the loss of the muscle bulk and the little twitchy muscles and the loss of reflexes. And the unique thing about ALS is that ALS, both of those things happen, right? And they happen in the same area of your nervous system. So you might have a limb that's got both spasticity and jumpy reflexes, but also little twitchy muscles and loss of muscle bulk. And so if you wanted to do that, if you wanted to say, where can I point my finger in this system that would do both of those things, where you'd point your finger is where these two things meet. Does that make sense? And so that's the anterior horn cells. That's that part of the spinal cord where these motor neuron cell bodies are. And that's really the area of the nervous system specifically in these pathways that is involved in ALS. And so it's a clinical diagnosis, and we make the diagnosis by seeing those signs and seeing this gradually progressive, painless weakness, upper and lower motor neuron signs, and not involving any of the other systems in our nervous system. And, when we look under a microscope, this is again, so this is that same kind of picture, right? This is the motor cortex, and this is those nerves coming down, and they're coming down into the spinal cord. They travel in the spinal cord in this thing called the cortical spinal tract, the cortex to the spinal cord. And then they go out and they touch the lower motor neurons and they go out to the muscle. And so if you look at a section of spinal cord, that's what this is here. This is a section through someone's spinal cord. And the cortical spinal tract in an ALS patient, maybe it's going to be pale, maybe it's lost some cells, right? So this would be a normal person. This would be maybe a person who's lost some of those cells. In other patients, or maybe in the same patient, if you look further down, so this is the part of the spinal cord where this nerve touches the lower motor neuron, goes out to the muscle. See these little blue dots? Those are these cell bodies of these lower motor neurons. And there's a lot more of them here in this control than there is here, because they they've died, right? So the lower motor neurons have been lost here. So this is the kind of thing that we would see under a microscope. If you look all the way out at the muscle, so this is a section of muscle. This is looking under a microscope at a piece of muscle. And this is what normal muscle looks like. It's sort of like a cobblestone pathway, and all the fibers are about the same size and shape. And when muscles lose their nerve, they become small and shrunken and atrophic and kind of angular and pointy. And so this is what we would see under the microscope to correspond to those clinical changes. So, what causes sporadic ALS? And as you guys well know, and as some people have already said today, we don't know. The causes of really what makes somebody have sporadic ALS is just unknown. About 10% of ALS or thereabouts is probably genetic. And this is a graph I should probably update it. This is from a publication a few years ago, but these are different genes, right, that have been implicated in causing ALS. This is that SOD1 gene, which was the first one that was discovered. This is C9orf. So the size of these circles relates to how common they are in the familial ALS. And this curve itself relates to how many genes have been associated with ALS. And I'll just tell you, between 2015 and today, this thing has gone up this way. So we've learned a lot more about these genes that can cause it. And it's a whole lot of different genes, right? That's a little bit unusual, but that's still only the familial cases. We still really don't know what causes most ALS. Now, one thing that we do know is that while it's the motor neurons that degenerate, there's a whole lot of other cells that if we look under the microscope or we look in, in our animal models or in lab, that are also goofed up, and they're goofed up in ways that seem to contribute to the disease. And maybe there's some problems in some patients, that start in the muscle or in cells called astrocytes or microglia. These are different, nervous system cells. And we know this obviously from looking at cell models and mice and, tissues of our patients who pass away. And, there's all these, this looks crazy, right? Pierre already explained, this is complicated science. So, and I'm going to blame, so this is actually Hristelina, who just spoke to you guys. Look at that. She wrote this back then. She's the author here. So this is her fault, right? I mean, look at how complicated this is. And so where does this stuff come from? These are all mechanisms, all different sorts of cellular pathways. And if you start, as I'm sure you guys have done, looking into this stuff, your head kind of explodes, right? I mean, where does all this come from? Why is it that all of these things are implicated in different types of ALS? And I mean, the simple answer is that, we're complicated. Our cells are complicated. Our bodies are complicated. And if you screw up one thing, a whole bunch of other stuff changes. So, you hear about a lot of different, what we call pathogenic mechanisms. So pathology means abnormal, right? And so pathogenic is something that causes a disease. And so these are the kinds of things, these are just some examples of, not in any way a complete list of the sort of things that people have implicated in causing ALS. And again, it kind of makes your head explode. And not just because it's a long list, but because a lot of these things are things that would affect every cell in your body, right? So why is it that this produces that all these diverse kinds of general cell mechanisms, how is it that they can produce this very specific disease where what happens is that we lose the motor neurons? Pardon me, my dog's barking. I think Amazon just delivered. And again, where does this list come from? How do we get to here? How do we decide that these things are important and that these are involved in ALS? And the way that we decide that, the way that basically we do, research is we have our disease and we have our normal, or maybe we have cells from our disease and cells from our normal, or a mouse that has our disease and a mouse that's normal. And we compare things and we look for things that are different. And sometimes it's obvious, right? It's pretty clear to tell that an apple is different from an orange. Sometimes it's not so obvious. This might be a different, variety of apple from this. I can't tell from looking at them. They might taste totally different. If I did an analysis on their chemistry, they might be different. Sometimes, you might only see a difference in some subtle laboratory experiment or in some assay, or even in some imaging studies. So here we got, these are actually MRI scans of, that's a peach, that's a pomegranate. So you got to be careful, right? Neither of those are apples, but pretty cool that somebody did that. Anyway, and so, the way that we look for treatments is by making these sorts of comparisons. And we can do that in cells. We can do that in animals that have genetic abnormalities that mimic certain types of the disease. We can actually look in tissues from our patients when they pass away, or different sorts of biofluids. And again, sometimes it seems like it should be obvious. So this is an experiment where, this is a treatment. These are mice that have a specific type of ALS, actually the SOD1 mutant mice ALS. These are 18-month-old mice. And these ones over here have been given a treatment of a drug. These ones haven't. And, you know, it kind of looks like these guys are moving around a lot more than these ones, right? You guys can sort of appreciate that. But it also should be clear if you look at it. So these are all litter mates. These are all mice in both pictures here that were born in the same litter from the same mom. And look how different they look. They're different colors, right? There's at least one dude over here that's moving around probably better than his other ones. A lot of these ones look paralyzed. And so I think you have a lecture later today on statistics and how do we know if something's significant. And this is a real problem. It's something we have to think about. And, you know, so Terry has been somebody who's done a lot of research on how do these models behave also when we breed them. And we've learned that just breeding successive generations of these mice or breeding them to different species change different things. So there's a lot of confusing questions. Even though this picture, if you looked at this, you might say, whoa, give me that drug. The mice over here, they're walking a lot better. You got to make sure, is it actually changing the thing you think it's changing? If you did something genetic to the mice, is it the thing that you think it is? So this is my house. I live out in the country in the middle of nowhere. We moved here in 2013. We're like totally off the grid. We have, you know, oil and well water. When we bought this house, the only reason we could afford it was that it was vacant for like 40 years and everything was broken. There were 52 broken windows. We had to replace all the plumbing, like the electric. It was a mess. Our families thought we were crazy. You know, it's got a big pool and my son loves it and whatever. So we fixed everything up and we were all excited and you know, we made it ours and we loved it and we're so happy. And then that year we had this unbelievably terrible winter in Baltimore or where we live up north of Baltimore. And, you know, we learned that this house that was built in 1940, that when the wind blows outside, the wind blows inside, okay? It is the draftiest, coldest, most freezing house ever. And so if you were doing an experiment, if you didn't know what made a house cold, if you didn't know what made a house, you know, drafty, and you just said, okay, let's compare my neighbor's house that's built like 5 years ago to our house and say, what's different, right? Can we figure out why my house is colder? You know, maybe the first thing you're gonna notice, I mean, we burned a lot of fires that first year, right? Is that we have a lot of ash in our fireplace. And if you didn't know what a fireplace was, or you didn't know how it worked, you know, you might accidentally perhaps conclude that, hey, that's the problem. We have to take the ash out of the fireplace. That's why the house is cold, right? And, you know, a lot of the things that we see when we make these comparisons in ALS are perhaps aggregates of proteins, things that don't belong where they are. And we have to question, we have to say, is this something that's causing the disease, or is it just something that's happening because of, you know, something unrelated? In the one that you guys might hear about a lot is a protein called TDP43, and it turns out it's not ash in the fireplace. It's something that normally is in the nucleus of a cell. And when there's this abnormality, it goes out to the cytoplasm and it gets stuck there and it can't get back and it clumps up. And so what happens is that it actually can't do what it's supposed to do in the nucleus. And that seems to be why it's involved in this disease. But the way that people figured that out was by examining all these different complex pathways and looking at normals and looking at the diseases. Now, let's say you understood how fireplaces worked and you knew, okay, that's because, you know, this was just generating heat. You know, we burned a lot of fires that first year, not since, because what we learned is that it makes it really warm, like 5 feet away from the fire, but it actually cools off the rest of the house, right? Because it pulls in drafts. And so, you know, anything that you adjust one way, adjust something the other way. This is our furnace. I think it's a good furnace. I don't know. I can guarantee you we need to service it a lot more than our neighbors. And we burn a lot more oil. This is our oil tank outside. This is a picture I took that first year. This plunger's showing us how much oil we have. It's supposed to be up here, right? So middle of the night, in the middle of winter, we're freezing our butts off because we have no oil because we're burning too much. If you concluded from this that we needed to burn less oil, and that was the difference between us and the neighbor, that would not be correct. However, you know, it is true that in essence, a lot of the pathways that we look at in ALS are pathways related to energy and metabolism, things like mitochondria, the powerhouse of the cell, right? Remember that from high school. You know, problems with energy utilization. And you might imagine the problem's not with the furnace, the problem's somewhere else. But if you looked at comparing controls and normals, this is a problem you might see. So what's the point? And I mean, I'm sort of laboring it, but, you know, our bodies, by adjusting these interrelated pathways, we're trying to compensate ourselves for the abnormality. And there likely are many different abnormalities in cells that can cause ALS. And in many patients, it might be the product of a lot of these things in the way that they interact. we actually still don't understand how all these general mechanisms and these diverse mutations can cause this very specific picture. And even more frustratingly, some of these same things are implicated in other disorders, right? So how does it decide that one person gets ALS and one person gets something else? Now, ideally, and this is the personalized medicine that Pierre was talking about, you'd like to address the specific underlying abnormality, like the genetic abnormality. What's the genetic abnormality in my house? We have these single, metal sash, single pane windows from 1940, and our house is made of stone and it's 2 feet thick. And we can't, genetic therapy is hard. We can't replace these windows. It's virtually impossible. But ideally, you'd like to address the primary problem that's causing, and luckily in ALS right now, actually things are getting easier, right? And as somebody mentioned, we have some therapies that maybe are promising in gene therapy. This is my father-in-law. And when my in-laws would come visit us, they'd go from room to room and turn the heat up, and we'd burn like $80 worth of oil every day. And we'd follow them around from room to room and turn it back down again, right? And it's totally driving us nuts. And this was our, one Christmas, we gave him these footy pajamas, so a side effect of this treatment was it made him look hilarious, but it actually was a good treatment, right? Like this didn't treat the genetic problem. This didn't treat the primary cause. This didn't actually even warm up our house, but he didn't feel the cold. It worked well. And this is not so different from some things that are in ALS trials right now. So some of you might have heard of tirasemtiv or reldesemtiv. These are medicines that they're not treating anything that we think is a cause of ALS. What they're doing is increasing the efficiency of the remaining muscle fibers and helping them to contract more efficiently. That picture I showed you of those mice, that's actually a project that I'm involved with that just has a preprint come out right now. And this, by the way, this is those mice walking around. And when you look at papers, and tomorrow Terry's going to talk to you about how to read a paper and how to critically read a paper. These are actually tracings of the mice walking around, and you can quantify, look, they walk less, they walk more. This is a treatment that, again, it's not something that we think is involved in the cause of ALS. It's actually helping the muscle membranes to repair more quickly, right? So, the muscles cells break down and they become atrophic when they lose their nerves. This is helping it's almost as if you had the cold temperature on because the air conditioning is on. Rather than turn the air conditioning off, you're just turning the heat up also. And I want to point out a couple of things. So these are the kinds of assays you might see in papers. So, you treat with the drug versus saline, and this is showing body weight. So the mice are retaining their weight, they're living longer. These are the kinds of assays that people look at in a mouse or maybe walking around. Again, we don't think that this is a cause of ALS. And by the way, I don't know that this works. And, this is what's called a preprint. Preprints are relatively new. This has not undergone peer review yet. And preprints are great because they're helping to spread knowledge very quickly and helping get ideas out there, but you have to understand how to read them critically, and you have to understand that they haven't been peer reviewed. And so I can't tell you if this works. I hope it does, but I hope everything does. I kind of think of preprints like when the car was first invented, and once upon a time, everybody was in horse-drawn buggies. And then like the first cars came out, right? And they were faster and they were better. And just like preprints might help you get information faster. But guess what? Like those first cars blew up. And, I have a 17-year-old son and he's in driver's ed right now. And holy moly, does he need driver's ed? Because you have to learn how to drive a car. And there's a whole lot of training that has to be done. And the reason I'm belaboring that point is it's really important that we learn how to read papers critically, and that, as research ambassadors and advocates, that these are skills that you develop, and you're going to work on it tomorrow some with Terry. Because while I think preprints are helping us to spread information more quickly, it's going to require some education and some training, much like it does to get a driver's license. So why have we not been so good at translating these findings from the lab? And I'm going to give an example here. Some of you might remember a ceftriaxone trial from a few years ago. This was, there was this specific mechanism, this glutamate transporters that they found in models of ALS and in patient brains were decreased. And it turned out that this antibiotic called ceftriaxone seemed to increase them. And it worked in the mice, and it worked in some early phase trials in people. It made the mice survive longer. And then it completely failed in a phase 3 trial of people. It didn't do anything. And so when it failed, we weren't sure why it failed. Like, did it fail because the hypothesis was just wrong? It was a stupid idea, or maybe it only works for a certain subset of patients. Maybe it failed because it actually didn't get where it needed to go, or maybe it got there, but it just didn't do what it was supposed to do. And the problem is with a lot of ALS trials, we don't know the answers to these questions. When a trial fails, we often don't know why did it fail. If we'd had a way to track the levels of that thing it was supposed to be increasing, maybe that we could have answered that question. And this is where biomarkers come in. And you guys hear about biomarkers a lot. There's actually different kinds of biomarkers. And I want to very point this out because it's an important thing to understand. So what exactly is a biomarker? So this is one instance right here. So you've got something you're measuring in a patient, maybe something in the blood, and in controls it's low, and in an ALS patient it's high. So if you had to name this kind of a biomarker, this is what we would call a diagnostic biomarker, right? It's something that's a different level in your patients versus your controls. That's actually not the kind of biomarker we need the most. We really don't do that bad of a job at diagnosing this disease. I mean, we'd like to do better, we'd like to do earlier, but let's give some other examples here. So, you know, there's trials right now of a treatment for a gene therapy for SOD1 ALS, right? You would like it if you're only enrolling patients who have that mutation in that trial, because it shouldn't do anything for anybody else. And so you have a test, that mutation test, that tells you who has that mutation. And that's a kind of a biomarker too. It's called a predictive biomarker because it predicts who's going to respond to the drug. And it doesn't have to be for a genetic mutation. Let's say that you have a drug that works on a pathway, inflammation in the brain, okay? You would like it if you had some sort of a test to figure out which ALS patients have a whole lot of that inflammation, because they're the ones you'd like to put in your trial. You know, we worry that trials may fail because we're enrolling everybody from everyone for a treatment that might only work for a subset of patients. So how about this one? So look at this. This is something that's getting measured, whatever it is, and measured one time. And if you have a high amount of that or a low amount of that, your survival is different. So a high amount of this thing, you tend to survive longer. This is how many patients are surviving over time, right? So 100% of patients are surviving, and then it gets less with time, or a low amount, less are surviving. Or maybe there's some specific function like strength in your arm or swallowing or speech. And again, you measure this thing one time, and over time, if you have a high amount, you do better. If you have a low amount, you do worse. So this is what we would call a prognostic biomarker, right? It helps decide maybe who's a fast or a slow progressor. And this is really important because ALS is so protean, because it can go fast or slow or affect different people different ways. And when you're designing your clinical trials, you want to make sure that you're comparing, again, apples to apples, that you don't have too many patients that are fast in one group or slow in another, because you won't be able to see through that noise. This is the one that we need the most. So let's say you've got something that you can measure, and each of these lines is a patient. Okay, so one red line is a patient. This is over time, and you measure something a bunch of times. And while there's variability, in every patient, it stays pretty flat. And then you give them a treatment that does something beneficial, and it decreases. Or maybe you have something that with normal time, it's going up, and then you give a treatment, and it stops going up. And so this is what we really need. This is called a pharmacodynamic biomarker. And the idea is, if you have a treatment that does a specific thing, let's say helps mitochondria, be more efficient, you need some way to measure to know, is it actually doing that? Did ceftriaxone upregulate that thing called EAT2? Ideally, we could have measured something and seen that when we gave treatment, it changed. And so these are the biomarkers that we really need the most. You have to be careful because we live in this world of big data, right? Where everybody's measuring everything from everyone, and there's a lot of correlations, and correlation doesn't prove causation. This is a really interesting website to play with. It mines publicly available data sets and looks for correlations. This is the divorce rate in Maine versus the per capita consumption of margarine in the US over this decade. Pretty tight correlation, right? I don't think anybody thinks that those two things are related. This is another one. I love this one. This is the per capita consumption of cheese in the US from the USDA versus from the CDC statistics on the number of people who died by becoming tangled in their bed sheets. Now, again, I don't think that these two things are related. There's 2 points I'd like to make about this. One of them is I kind of like the fact that in this country that the appreciation of cheese is increasing because I love cheese. I also think that this bed sheet thing is because kids don't learn how to make their beds anymore, and maybe that's our fault, but my kid, forget it, he can't make his bed. But again, you have to just be careful. And so while a biomarker, might be something that measures something important for a disease, you kind of got to prove it. We actually have a whole lot of promising biomarkers. If you type biomarker and ALS into a search engine, you'll get a bazillion things. And the question is, what do we need to do once we think that they're promising to make them clinical trial ready so that we can use them in clinical trials to improve clinical trials? this is one example. You might have heard of neurofilaments. It's one that is getting a lot of press. This was a study that we did. And what you have to do is you have to try measuring them in a bunch of different commercial assays, in a bunch of different labs, in a bunch of patients. You have to figure out how do the different assays perform, right? Do the samples measure the same in different people's hands, in different labs, in different centers? What's the statistics on these various assays that are available? And that's not sexy stuff. That's not figuring something out, but it's important stuff. And so that's exactly what this study was. So this used the samples from the CREATE consortium, which is a big consortium that's collecting all of this wonderful longitudinal data and biofluids. And look at this, we took the same serum, the same spinal fluid, and we sent it to different labs that ran different assays. And we said, how do these assays compare to each other? Because this is the kind of information you need to be able to use it in a trial. And in this paper, we talk about them as a prognostic and potential pharmacodynamic biomarker. This picture you might see kind of looks like the one I just showed here, where you measure something and they sort of separate from each other over time. And that's exactly what this is, right? So if you had a low level of this thing or a high level of this thing, and you looked at survival over time. So this particular graph is showing you that maybe this is a good prognostic biomarker. So, the biggest problem and the thing we worry about the most are these animal models and cell cultures and all these things we use in the lab. Do they really matter for sporadic ALS? Are they a good model? And over the past couple of years, and I think Pierre said that, it's the ultimate model is the iPS cells, or we think that these assays that we have, these tools that are based on patient tissues and patient biofluids and patient cells are maybe the answer, that maybe validating these things in those types of assays is going to help us. But you have to be careful still, right? Because if you're looking at something in a cell culture, let's say that you've got, as Pierre was mentioning, iPS cells that you make into neurons. Well, is that really, so if the iPS cells are apples, these neurons, our brain is kind of more like apple pie. And if you do something where, there's a lot of other stuff besides the apples in our brain, other kinds of cells, other stuff going on. If you take those cells and you grind them up and you measure some chemical in them, that's kind of more like making applesauce. And maybe that's not the same thing as what's going on in our brain. So it's important that we use these tools correctly. And it's also important that we have very rigorous clinical and pathological data so that we know that we're getting valid answers. And in fact, that was where I just was for the past 2 hours, was giving a different webinar all about these sorts of biorepositories and how important they are. And the main point of this webinar, there were 2 points. One of them is we can't do any of this stuff without our patients. And we are so tremendously grateful, because without you, we don't get these samples and we need them. And we think that they are going to be something that really helps us bridge this valley of death and figure out, why things aren't translating. And, the other part of this was a talk to the researchers to make sure these things are being used responsibly, because if you donate tissues or your honestly to our research, that's the ultimate donation, and we need to make sure we're doing everything we can to make sure that they're used responsibly. So that's where I was. And by the way, back to that preprint, what's my role in that study? My role is that I direct a postmortem tissue effort, and those mice that I showed you, this is a measure this is a stain of something, that this is a normal mouse. This is the ALS mouse with the SOD1 ALS. This is this abnormal stuff. Same thing here, normal mouse and ALS mouse. These are pictures of their muscle. And my part of the study was that, we have these tissues from our patients, and we wanted to see, could we see the same thing? And indeed we did. So looking at patient muscles, and look at this, the mouse was an SOD1 type of ALS, but we looked at sporadic ALS. We also looked at C9orf, right? And so we see similar things. Now that's encouraging. It suggests maybe this is something that might work for different kinds of ALS, but maybe not. And I would ask you to please, just like I'm hoping my peers do, critically read our preprint, like you might critically read all of them, and look for ways that we might have got this wrong, or more work that we might have to do. So how can we think about this biomarkers earlier in the process? So one way is teaching the next generation of scientists. And I lecture to the grad students at Hopkins who are early on and just learning about what they're going to do with their lives. And I lecture them about ALS. And this is the test question that I give them. I don't ask them about like scientific minutia. I say, hey, I want you to choose some mechanism or actually propose a new mechanism. I don't care. Design an experiment, right? And not just design an experiment, but tell me what you would use as a biomarker and how that biomarker might be carried through to an eventual clinical trial, because this is what we need. We need in the very earliest types of research, basic science, to be thinking about how are we going to make biomarkers for clinical trials. Another way that we can encourage this, and we're going to talk about the DoD program tomorrow some, is in fact making biomarker development and inclusion a requirement for grant mechanisms. These are some of the DoD ALS research program mechanisms, and even the earliest mechanisms, these hypothesis-driven stuff that is very early on. We give people more money if they have a biomarker development in parallel. We require it for these other preclinical awards. Tomorrow, we're going to talk more about that program, because I'll tell you what, the reason we do those things is because of people like you and industry and all of the voices in the ALS research community that we surveyed and said, what do we need to do a better job? How can we make this faster, better, more effective? And so we're going to talk about that some more tomorrow. That's all I've got.
Thanks so much, Lyle. Appreciate it. Does anyone have any? Yes, thank you, Jinsey, got it started. Does anyone have any questions for Lyle? Do we have anything in the, nothing in the chat box, but now's the time. You can unmute or write a question in the chat box. Yes, no?
It's late in the day.
I got a question.
Yeah.
When, we talk a lot about biomarkers, and we have like consortiums bringing in samples, and, you have a lot more experience about collecting those samples and cataloging them and then having to redistribute them. What are some of the challenges in trying to maintain those? And, is it hard to get funding for those types of things?
Well, so, I mean, honestly, it's an evolution, right? And so there's a lot of reasons. In a perfect world, you are going to exactly meet the needs of your researchers. Now, let's be honest here. As I started off by saying, we've done a terrible job in ALS, right? Our clinical trials fail. So I honestly can't tell you what's important until something works, right? I mean, I honestly don't know what's a good biomarker, because we don't, in fact, really know too well, how to choose these things. So the best we can do is make sure that the samples we're collecting meet the needs of our researchers, that if there's quality control measures or different kinds of tissues, today somebody wants a certain region of the brain to look at C9. 10 years from now, it might be something totally different. Today, somebody wants frozen tissue. 10 years from now, they might want tissue that's been, I don't know, soaked in, beer. I'm not right. I just don't know. And so what we try to do is standardize the way that we are polling the research community to make sure that we are trying to meet the needs of the most researchers possible with the resources we're providing, and doing it in the most efficient way. The biggest barrier is some of the regulatory and issues, with just sharing data, sharing samples. It's a little easier for postmortem tissues than it is for biofluids. It's a moving target. It's something we're getting better at, but it's hard work. And I'll tell you, it's hard work that I love to do, and because we're so grateful to have this valuable resource, and I do think it's going to make a difference. So one last thing I'll just say is that if there are patients or family members who have reservations or questions about this kind of thing, there's certain ones we get. One is, oh, if I donate to a biorepository, that's a clinical trial. So now, my clinic's not going to let me enroll in the other clinical trial. They're going to give somebody else a turn, right? That's just not true. There's not a clinical trial anywhere in the world that having contributed biosamples makes you not be allowed to enroll in. And in fact, more and more clinical trials are including biosample collection and analysis as part of what they're doing. I'm happy to talk to anyone, anytime, who has questions about postmortem tissue donation and answer any questions about that.
Oh, okay.
I think
I have.
Oh. Go ahead, Christina. I can speak after you.
Oh, I was just going to ask if you donate your body when you're gone, how you go about that.
Yeah.
Or if you need, oh, go ahead.
No, go ahead. What were you going to say?
Or if you need tissues now, like a muscle tissue.
Right.
I'm willing to donate.
Yeah. So I got to tell you, patients, I'm in awe of how amazing all you guys are and the stuff that you do for this research community. And we wouldn't be able to do anything without you. There are these established programs, and I'm going to talk about them tomorrow, because actually we have a database of them as part of the DoD website that talks about all the different resources that researchers can get to. There's different efforts at different centers that are collecting tissues. We all talk to each other. We all know each other. We all work together to get researchers what they need. It depends on where you live. And if you have questions, I'm happy to talk to you about it. It also, having an autopsy in this country is something that your family has to actually consent to after you die. And so because of that, it's something that's important to talk to with family and you know, and loved ones. If you consent today and say, I want to donate my brain to ALS research when I die, but then after you die, your family says, nope, that doesn't happen either. So, you know, it's important to talk.
What I was going to say is kind of a link between what Jincy and Christine mentioned. I do know that, you know, Piera is on probably the list that Lyle has. So Piera organized a biobank for the Jefferson University. So she can probably tell you a lot how difficult in terms of funding it is. It's not an easy thing. And also Christine, also you can talk with Dr. Passinelli about this.
And last thing just to mention, so we have a new effort at Johns Hopkins that's evolved from the one that we've been participating in. Our new effort is a collaboration with the CDC ALS registry, and starting, I believe in October, we're actually going to be able to accept tissues and data from any academic center in the country, and we're going to be able to train them according to our very strict SOPs for data collection and dissection so that we make sure that everything's done the right way. So.
Yes, and I would echo, it is very difficult. We have a tissue bank as well and costs a lot of money. You've got to have freezer space, you've got to have maintenance, alarms, just collecting the tissue. You know, usually somebody who manages it, make sure that the samples are collected properly. We like, and I know Piera does too, we all have uniform collection procedures to make sure that we're all collecting things the same, because if I collect serum and put it at -20 and Lyle's putting it at -80 and, you know, Piera's leaving it at room temperature of 4 degrees, that's, you know, you can't expect similar results. So we have to have SOPs, standard operating procedures that are shared. And
the kind of tube that you put a biofluid in seems to matter. How much airspace is in the tube seems to matter. You know, and when I say matter, maybe it doesn't matter because maybe we're not measuring the right thing. But what I can tell you is the sorts of assays that we do on these samples, it changes them. We spend a lot of time trying to figure out how we can screw up an experiment, right? We take a test set of samples and we say, okay, I'm interested in this particular measurement. I want to know how I can goof it up before I waste 200 samples, right? So if I leave it out on the counter for a day, what does that do? Or if I, you know, put it in a different concentration of salt or whatever it might be. And that's the way we learn about these things.
And how long can we keep testing the same sample for something? How many freeze thaws can we do?
Yeah. Well, so we figured out, for example, that it's better not to do freeze thaws, right? And so instead we divide these samples, whether they're fluids or tissue, into itty bitty pieces that are the exact sizes that researchers need for the kinds of experiments that they're doing. And we do that up front. And that way we can just go get that piece and send it to them without having the freeze thaw. It's a perfect example. I don't want this, you know, this wasn't supposed to be a talk about this kind of thing. So, but obviously I talked too much.
But somebody brings up and it does take a lot of standard operating procedures and a lot of work to maintain a tissue bank. And it's not something that's easily funded.
But tomorrow,
I would like to add to that for all of you who are in the Philadelphia area and the tri-state region, right? That following what Terry was saying, we really try to coordinate. As of now, Jefferson is really doing tissue bank and collecting post-mortem tissue, not only from Jefferson patients, but also we and Terry and us, the LS Center of Hope and us really collaborate, also patients outside Jefferson, precisely because of that, because we have to follow standard operating procedures, because we have to standardize everything, and because of the cost. So if we can work together on that, it's really better for you and it's better for everybody. And the quality is, because as Lyle said, this is really the ultimate gift that you give us, and we want to operate with the best way possible and coordinate. So as of now, our tissue bank program is really open to Jefferson and non-Jefferson patients.
So, and just a little pitch for tomorrow too. So tomorrow, Terry's going to talk to you about how to critically read a paper. And then we're going to talk about a little about the DoD, the ALS research program and the involvement of advocates. But specifically, one of the feedbacks we've heard from advocates is that we need to teach you guys how to read a grant proposal, right? We need to teach you guys how to assess, because you read the impact statement and it says, this is going to be great for ALS, because let me tell you, every scientist I know can write an awesome little sales pitch about whatever they're doing. And so we've realized that we need to have a formal curriculum to teach research ambassadors and consumers, because really we work for you, right? And we need your opinions, and they need to be the right educated opinions. We're going to talk a little bit about it tomorrow, but in the future, we have full plans to roll out, in essence, a curriculum to help train you to evaluate research proposals. And other CDMRPs have been very successful with this, so we're not reinventing the wheel. We're kind of learning from them. We'll talk about it more tomorrow.
Very good. So I think we'll take a 10-minute break, and then we'll start at 5:32 on the Taskmaster. Jinsey, you're all set to go. So we're really looking forward to it. So everybody take a little break. If you have something to eat and want to eat during this, by all means, if you feel like you have to turn the camera off to eat, that's no problem. But we're looking forward to Jinsey's talk. We'll see you in 10 minutes.
Hey, Terry, do I, what time does the talk after me start?
Oh, 6:30.
Okay.
You can go as long as you want, because Eric is taking his father to the doctor. He says he'll get on as soon as he gets back. So
Terry told me I had 2.5 hours, so blame her. It's not my fault.
well, you did. You did have 2.5 hours, Lyle.
Oh, look, he's muted. You're muted, Lyle.
I said whatever it takes, right? That's important,
That's right.
Very good.